Okay, morning all. This is the first revision lesson type thing that I'm going to do as a request. Somebody requested this from my class. It's on transport across membranes, and it relates to booklet 2, cells and cells transport, towards the end of that booklet. Beginning with the cell surface membrane. So we'll look at the different types of transport across the membrane, try to explain why each of those methods of transport are or occur, give some examples of the type of molecules, and help you with your explanations. And then at the end, there are a couple of typical sort of exam type questions that you might see relating to graphs. So, to begin with, it's probably worth just reminding ourselves of the cell membrane. So, cell membrane is mostly composed of these phospholipids. And if we just remember, a phospholipid is a glycerol molecule. So that's 3 carbons. With, how am I going to draw this? Okay, with 2 fatty acids. And that zigzag line represents a hydrocarbon chain. Okay, and I'll draw another one there. And at the end of these, now, unlike a triglyceride that would have the glycerol and 3 fatty acids, one of the fatty acids is replaced by a phosphate, an inorganic phosphate group. Okay, that's an inorganic phosphate, and the inorganic phosphate has as a charge. Okay, it's slightly negative. So this makes the phospholipid polar. So this region of the phospholipid is hydrophilic. Because we know also another feature, another really important molecule when we think about this is water. Water has a structure something like this. It's got a couple of spare pairs of electrons up there, and oxygen is electronegative, so it has a pull on this pair of electrons that's shared between the oxygen and the hydrogen. So those electrons are in a higher density closer to the oxygen. That gives the oxygen a slightly negative charge. And because the hydrogens, well, they've not lost, but the electrons have been pulled away, leaving just the proton inside the hydrogen atom here, that gives the hydrogens a slightly positive charge. Okay, so we refer to water as being a polar molecule. It's got this slightly negative end and a slightly positive end. That means it is attracted to other molecules with charges. So, for example, we've got this inorganic phosphate molecule here, or inorganic phosphate group on the this is a phospholipid. Okay, that inorganic phosphate has a slightly negative charge because it has oxygens which are temporarily, well, it changes, but they those oxygens have a slightly negative charge. Anyway, so the water, the positive or the slightly positive pole of the water is attracted to that slightly negative pole on the phosphate group. Hydrocarbons, on the other hand, so this region of the phospholipid are non-polar. So refer to them as being hydrophobic. And that's because, well, this is just an example, a really simple hydrocarbon. This is methane. Okay, the electrons are shared equally between the carbon and the hydrogen. Neither of these atoms are electronegative, so there is no charge on this molecule. And that makes it hydrophobic. It means that it will repel any polar molecules such as water. So or across the membrane of a cell, we've got this phospholipid bilayer. I should have done this first, really, shouldn't I? Okay, with the hydrophilic heads, this region, facing outwards towards these aqueous solutions, towards the watery solutions that are either on the outside of the cell or on the inside of the cell. The tails, hydrophobic fatty acid tails, which are on the inside, face each other, and then they form a bilayer. So typical question might be to explain the appearance of the membrane, something like that, and you would say that the hydrophobic fatty acid tails face each other, hydrophilic phosphate heads face outwards, and it forms a bilayer, something like that. Okay, so because of this hydrophobic region in here, between where those tails are present, it's a hydrophobic region, any polar molecules are unable to cross the membrane. Now that's not completely true, but let's use water as an example. Now water can, it doesn't do it very quickly, can just about pass through this, can pass through the membrane. But let's just say, for the sake of argument for now, that because of its polarity, it's unable to. And in fact, it doesn't do it very easily. Another example might be something like an ion. So a sodium ion definitely cannot. Okay, so it's unable to pass through that hydrophobic fatty acid region between the bilayer. Okay, things that can are non-polar. So molecules such as oxygen, I don't know why I've done that in a circle. Ooh. So oxygen, for example, which is non-polar, can freely pass between these hydrophobic regions, because that's non-polar, hydrophobic region is non-polar, so therefore it's able to get across. Okay, other things we might describe as being lipid soluble. So lipid soluble, or things that are composed of mostly hydrocarbons, can also pass through that hydrophobic region. Okay, so essentially charged ions and molecules with polarity do not pass through. Water does a little bit, but not particularly well. Anything that is uncharged, so any non-polar molecules can pass through as long as they're small enough. Larger molecules, we can just have a look in the notes here. So larger polar molecules such as glucose or other things are unable to pass through. So small non-polar can pass through. Hydrophobic molecules, so lipid-soluble or hydrophobic molecules can pass through. And if they are small enough, I suppose, let's say that water can move through, but doesn't do it particularly well. Ethanol as well. Ethanol is a lipid-soluble molecule, and that's able to pass through. Right, there's a little bit of background. Let's have a look at the types of transport then. So the first type is diffusion. So what is diffusion? Well, here is the definition. So it's the net movement, and by net we mean net movement means that there is movement in both directions, but the overall movement is what we refer to as the net movement. So these, if we have a look here, these particles could be moving in any direction. In fact, they are moving in any direction. But there is a higher concentration at this end compared to a lower concentration at this end. And overall, the movement of those particles will be from the high concentration towards a low concentration until an equilibrium is reached. So an equilibrium is where there is no concentration gradient. No concentration gradient. And the reason that that diffusion stops when equilibrium is reached, well, again, those particles are moving in every direction, so they will be moving back and forth, but there's no net movement when equilibrium is reached. Okay, this is passive, which means that there's no energy required from the hydrolysis of ATP. And the types of molecules that can do it are oxygen and carbon dioxide. So sometimes we refer to this as simple diffusion. So going back to my example here, oxygen, carbon dioxide are both non-polar. They can simply diffuse through the phospholipid bilayer. Okay. So the molecules oxygen and carbon dioxide will simply or move by simply diffuse through the phospholipid bilayer. Okay, or across the phospholipid bilayer. Brilliant, yeah. So simple diffusion across the phospholipid bilayer. The next is facilitated diffusion. Okay, and this is different. We could say net movement again. So the net movement of molecules that cannot move by simple diffusion from a higher to lower concentration. So again, it's from a high concentration to a low concentration. But these molecules are unable to move by simple diffusion through the phospholipid bilayer. It is passive, but it does require some form of protein. So, dotted throughout the phospholipid bilayer are channel proteins and carrier proteins. Okay, so channel proteins allow for the facilitated diffusion of charged particles, ions, such as sodium ions or chloride ions or potassium ions, etc. Okay, in neurons, sodium ions and potassium ions, and I suppose chloride ions, will move by facilitated diffusion across a phospholipid bilayer from a high concentration to a low concentration, but via a channel protein. So the difference between simple diffusion and facilitated diffusion is the necessity for one of these channel proteins or a carrier protein. We'll have a look at carrier proteins in a minute. Okay, so the pore size, so if we imagine this is the pore here and here, will determine which ions can pass through. And there are also charges on the inside of the channel. Okay, and those charges will be related to and will determine which ion is able to pass through. So, for example, here this is a sodium ion channel. The amino acid will have R groups, which are negatively charged, and they face towards the pore, towards this channel, and then that allows the sodium ions to move through. Okay, so facilitate diffusion via channel proteins. These are the ions. Facilitate diffusion by carrier proteins is slightly different. So, still another protein which crosses the membrane, crosses that phospholipid bilayer, but these transport water-soluble or polar molecules. Okay, for example, glucose and amino acids both require carrier proteins for transport across the membrane. Again, from high concentration to low concentration, so this is still passive, but this time with carrier proteins. Carrier proteins if they are water soluble. Water soluble. Okay. The difference between carrier proteins and the channel proteins is that the carrier proteins change shape, whereas channel proteins have a fixed shape. So, the shape of the carrier proteins is specific and complementary to the molecule which they transport. So, if we can imagine this is glucose, the shape of the glucose is complementary to the tertiary structure of the carrier protein. Glucose enters that complementary shape. When it enters, that causes the carrier protein to change its tertiary structure, and in changing the tertiary structure, they transport that glucose molecule across. And then we would say that the glucose is diffused across the membrane via that carrier protein. Okay, so fixed shape with the channel proteins, and then the tertiary structure changes in. So fixed shape with the channel proteins, and the tertiary structure of the carrier proteins changes, which allows a specific molecule to pass through. Okay. Next is active transport. So, active transport involves this idea of pumping. Okay, pumping there in apostrophes. It's to represent there that it requires energy. Okay, so Energy is provided by the hydrolysis of ATP, and that ATP tends to be produced by aerobic respiration. So, in cells that do a lot of active transport, we tend to find a lot of mitochondria. So cells that are adaptive for active transport will contain a high density of mitochondria. Okay, so this process is slightly more involved, a bit more complex. So again, it involves carrier proteins. Now in active transport is only carrier proteins. The tertiary structure of that carrier protein is complementary to the molecule which is being transported. So there will be a specific carrier protein for one specific molecule. Energy from the ATP. So when the ATP is hydrolyzed, the phosphate group becomes attached to the carrier protein. So ATP undergoes hydrolysis to produce ADP and an inorganic phosphate. That inorganic phosphate attaches to the carrier protein, and we refer to that as activation, or activating the carrier protein. Okay. At which point the molecule which is being transported is able to attach. So we get hydrolysis of ATP, the inorganic phosphate binds to the carrier protein, and then the molecule can attach. When the inorganic phosphate is released, okay, that we can see here, the energy provided by the breaking of that bond allows for the change in the shape of the carrier molecule. And as that carrier molecule changes shape, it moves the it moves the molecule across the membrane and from the area of low concentration to an area of higher or lower concentration to higher concentration. So I'll just go through that again because we had a bit of an interruption there. Okay, active transport requires a carrier protein. Carrier protein will be complementary to, or its tertiary structure is complementary to the molecule that it is transporting. ATP is hydrolyzed. The inorganic phosphate binds to the carrier protein, so meaning that the carrier protein is activated. When that inorganic phosphate is released, that provides energy to the carrier protein, causing it to change shape and move the molecule from an area of lower concentration to a higher concentration. Okay, here's a little example of where active transport might take place. So this is cardiac muscle. These are the mitochondria in the cardiac muscle, and these lighter pink stripy things are the muscle fibers. Okay, those muscle fibers are made up of a couple of proteins called actin and myosin, and to move over each other, they require ATP. So the mitochondria in that really high density can do aerobic respiration. That aerobic respiration provides the ATP. The ATP is then hydrolyzed during the muscle contraction. And then when the muscle contraction is finished, there's also ATP required for active transport. Okay. So if you see a question relating to a cell perhaps, or some kind of microscope image, there is a high proportion of mitochondria, it's likely that those mitochondria are there for active transport. Other examples might be in the small intestine. The uptake of glucose and amino acids requires active transport in co-transport. I've done another video on co-transport if you want to look for that. But the sodium-potassium pump in co-transport requires ATP as a form of active transport. The absorption of mineral ions in plant roots, another form of co-transport, but that is again from an area of low concentration to high concentration. And then in the neurons and the muscles, we have this exchange of sodium and potassium ions, again from low concentrations to high concentrations. All of those cells have high densities of mitochondria. Okay, so I'm just going to flick through to some factors affecting those rates. So diffusion, okay, if you've been in my lessons, you know I've banged on about this a lot. Fick's law isn't actually present on the specification anymore, but it's really, really important, and so much of the lower sixth biology depends on an understanding of Fick's law. So it states that diffusion is proportional to these three factors. So surface area multiplied by the concentration difference, or the gradient, divided by the thickness of the exchange surface, or the distance of diffusion, or the diffusion pathway, something like that. Essentially, if you increase the surface area, increase the concentration difference, and you minimize the distance of diffusion, the rate of diffusion will increase. So for a fast rate, large surface area, large concentration difference, and a thin exchange surface. Okay. Other things that might have an influence, things that might change the permeability of the phospholipid bilayer, allowing more or less simple diffusion. So unsaturated fatty acids have fatty acids that are more kinked. There we go, they've got two fatty acids that are kinked because they're unsaturated, and that means not on this particular one, that those phospholipids are further apart. Saturated fatty acids have much straighter chains and therefore fit much more closely together. So a membrane with a high proportion of unsaturated fatty acids would be more permeable. Other things that might change the permeability related to facilitated diffusion are the number of carrier or channel proteins. So a higher number of carrier or channel proteins makes the membrane more permeable for facilitated diffusion. Here's a little question related to that. Explain the evidence from the graph that this substance is entering the cell by facilitated diffusion. Well, we can see initially, as the external concentration of the substance increases, so does the rate of entry of the substance into the cell. So at this stage, between here to here, the limiting factor, or the factor which determines the rate, is the external concentration. At this point, the rate no longer increases. And that is because between here and here, the number of carrier proteins, or perhaps channel proteins, is limiting any further rate. And that is because those carrier proteins or channel proteins are transporting the substance at their maximum rate. If we imagine, the analogy I often give is if you've got a football stadium with only one turnstile open, the number of people trying to get into the football stadium will be very slow. As each person enters a turnstile, the football stadium will fill up really slowly. If you then increase the number of turnstiles, more people can get into the stadium, it would fill up quicker. That's essentially the idea here. We can say at this point that the carrier or the channel proteins are working at the maximum rate we can say that they are saturated. Okay, and therefore, if you increase the number of carrier or channel proteins, the rate of entry will continue to increase. Okay, active transport can be affected by this idea of the number of carrier proteins again, but also by factors which slow or speed up the rate of respiration. So low temperatures means there is less kinetic energy. With less kinetic energy, there will be fewer enzyme substrate complexes formed, which would mean that the rate at which ATP can be produced in respiration would decrease. There's less ATP, there will be less active transport. Likewise with oxygen. Oxygen is necessary for aerobic respiration. It acts as the terminal electron acceptor. If there's little oxygen, the rate of aerobic respiration would decrease, and there will be less ATP. This is a really good one to look out for. Some kind of respiratory inhibitor. That means it's stopping respiration. So again, less aerobic respiration. All three of these being there is less ATP. Less ATP or no ATP. So in your responses, you say less or no ATP, and therefore the rate of active transport decreases. On the other hand, what would increase the rate? Well, more mitochondria, higher temperatures, both related to respiration, so therefore more ATP, or more protein carriers, more carrier proteins would increase the rate of active transport. Okay, I hope that was helpful. If you have any more requests, remember to send me an email and I will do my best to talk you through that. Thanks very much.